name. So it's Einar. Yeah. Sorry. So my name is Einar. I'm currently a postdoc in School of Math at the University of Minnesota. Today I'll talk about phenotypic switching, and this is one of my PhD projects, which was done with Jasmine Fu and Kevin Leader. Now, as we've been talking about a lot in the last 2 days, cancer cell populations usually display a very high degree of heterogeneity, and this can be both due to genetic mutations and non-genetic or epigenetic modifications. And just to take one example, in certain breast cancer cell lines, 3 distinct non-genetic phenotypes have been identified based on the expression of the 3 markers that are shown there. But very importantly, these kind of non-genetic phenotypes can be reversible, meaning that they can sort of dynamically emerge from one another. And a common way to experimentally interrogate this kind of dynamic heterogeneity is to take a parental cell culture, use genetic markers, morphology, for example, to sort the parental cells into distinct subpopulations, and then you allow the subpopulations to expand over time. And what people have observed is that when you do this, the other phenotypes will emerge, so they will pop up in the population. And if you run this long enough, the population will eventually converge to sort of an equilibrium distribution, independently of which state you start with. So if in these experiments you want to understand the dynamics quantitatively, you can collect data at specific time points. And often people will collect data on the fraction of cells in each state at each time point. But if you can also measure the total population size at each time point, then you will get the number of cells in each state at each time point. So I will refer to cell fraction data and cell number data to distinguish these two. Now, Gupta et al. Is a paper maybe some of you are familiar with from 2011. They hypothesized that this kind of dynamic heterogeneity could be explained by a very simple Markov chain model, where cells are able to switch phenotypically between states or types. And our goal for this talk and in this work is to develop a framework to estimate both these rates of phenotypic switching, but also the rates of cell division and cell death. And when we develop the framework, we emphasized deriving confidence intervals for the model parameters, else I'll get into in a little bit. But just in case you're not convinced that this is a worthy question to consider, I want to talk very briefly about how this kind of estimation framework can be useful in the context of treatment. And so many recent experimental works have indicated that anti-cancer agents can actually induce switching from drug-sensitive to drug-tolerant or resistant cell states. So in that case, the switching rate mu there would actually change or increase as a function of the drug dose. And other more mathematical papers have indicated that depending on the level of induction, so depending on how much the drug affects the switching rate into tolerance, the optimal dose under continuous therapy, for example, can be much lower than the maximum tolerated dose. So I would argue that understanding the rates of phenotypic switching, understanding the rates of cell division and cell death for a particular cancer, both in the presence and absence of therapy, is crucial for understanding how to best treat the cancer. And I think here it becomes apparent why we're interested in uncertainty quantification or confidence intervals. Because if, for example, the optimal dose is very sensitive to the exact level that you get up to, so the exact level of drug-induced resistance, then you obviously have to know how confident you can be in the estimation before you can make any definitive treatment recommendations. Okay, but now I'm going to talk a little bit about the ideas behind the estimation framework. So here we're going to model the cell population dynamics using a multi-type branching process model in continuous time. And in the talk, we're going to assume that we only have 2 cell types, but in the paper, the accompanying paper, we did the more general case of 2 or more types. So this is a stochastic model where the 2 cell types, they're going to divide stochastically at rates B1 and B2. They die at rates D1 and D2, and they switch between states at rates nu1,2 and nu2,1. And we're going to let lambda1 and lambda2 be the net birth rates of the 2 types. And for the purpose of the talk, we're going to assume that we always start out with these isolated subpopulations. Next, we need a little notation. So we're going to let CJTN, this is the vector of cell numbers at time t in the model, when we start with n cells of type J. So C1JTN, so the first coordinate of the vector, this would be the number of type 1 cells at time t. and delta JTN, this is the corresponding vector of cell fractions. So delta 1 JTN is the fraction of cells in state 1 at time t. Now, in this branching process model, the individual cells behave independently, and this allows us to derive central limit theorems for both cell numbers and cell fractions at a given time. So more precisely, if we fix a certain time point, we send the starting population size to infinity, we can approximate the distributions both of cell numbers and cell fractions by normal distributions. And I should say here that, so in these experiments we were talking about in the beginning, it's very common to start out with hundreds, thousands, even up to millions of cells. So it's very reasonable to assume that n is large in this case. But essentially, the estimation framework is based on these approximations. So once you have these normal distributions, you can derive confidence intervals, and we simply use a maximum likelihood approach to get parameter estimates, and we also use the likelihood functions to get confidence intervals. But in the interest of time, I'm not going to go into details, and instead we're going to talk about some of the estimation results. So the first thing we did was to apply this framework to computer-generated or simulated data. So in the figure here, if you look at the bottom, you can see the 6 model parameters. You have first the switching rates, then the net birth rates, then the birth rates. The blue dots are showing results using cell number data, the red dots using cell fraction data. And so what we did is that we generated 100 different parameter regimes, so 100 sets of values for these 6 parameters. And each dot in the figure represents one parameter regime. And the position of the dot on the y-axis tells you the error in the estimation for that parameter under that parameter regime. So sort of more informally, the lower the dots are on the figure, the more accurately the parameter is estimated across these 100 different regimes. Okay, and so we can derive several interesting insights from this figure alone. So notice first that for all of the parameters, they're estimated more accurately when you use cell number data than cell fraction data. If we just look at the cell number data, we see that the switching rates and the net birth rates are estimated very accurately. The birth rates are not estimated as accurately, but still reasonably in many cases. For cell fraction data, however, we can estimate the switching rates pretty well. The difference in the net birth rates, so lambda 2 minus lambda 1, is estimated worse, and actually a lot worse in many cases. And the remaining parameters, so the net birth rate lambda 1 and the birth rates, we can't estimate those accurately at all in any of the parameter regimes. And we actually, in the accompanying paper, we also did identifiability analysis, so more theoretical, which kind of confirmed all of these numerical insights. But one very important takeaway here is that if you're interested in estimating both switching rates and growth rates, and even if you just want the difference in the growth rates, this lambda 2 minus lambda 1, you really have to use cell number data as opposed to cell fraction data. And actually, as we were writing this paper, we were contacted by 2 different colleagues who were thinking about this exact question. So what parameters you can estimate depending on what data you have. So I hope that this is a useful and practical insight. But so moving on, we also see in the figure that the birth rates, when we use cell number data, we can't estimate the birth rates as accurately as the other parameters. And we wondered whether we could add some kind of data which would make the birth rates more identifiable, more easy to estimate. And to make a long story short, if you add data on the number of dead cells at each time point, and so here you only need the total number of dead cells, you don't need to know which cell type is dying. In that case, the birth rates become as easy to estimate as the other parameters. So you can see in the figure here that when we add data on the number of dead cells, that doesn't really change estimation of the growth rate or the net growth rate, but the birth rate estimation improves a lot. So the takeaway here is that if you can add this data, the number of dead cells, you can actually estimate all of the parameters accurately. And finally, I want to mention briefly that we also applied this estimation framework to real cell fraction data. And you can see here confidence intervals for the 2 switching rates and the difference in the growth rates. And this example really further highlighted how difficult it is to estimate even differences in growth rates, let alone the growth rates themselves, if you're using cell fraction data. And so sort of an easy way to think about this is that if you're using cell fraction data, you may only be able to estimate the switching rates. So you may not be able to say much about the actual growth. Okay, but so with my final seconds, I'll say here that There are, of course, limitations to the method. We first of all assume that we have a population with discrete phenotypes, which can be identified using known biomarkers. We're also assuming that these cells behave independently. There's no interaction with other cells in the microenvironment. There are no spatial or nutrient constraints. But despite this, I still argue that, so this is sort of a useful work to understand what parameters you can estimate accurately, depending on what data you can get. And I would also argue that this is still useful for many examples in the literature where this kind of simple non-interaction model seems to work well. Okay, so since I have you all here captive, I'll promote my own work. So this paper that I talked about today was just accepted into the Journal of Theoretical Biology. Actually, the final version came online yesterday, so you can check that out. A couple of years ago, we wrote about how kind of switching between drug sensitivity and drug tolerance affects the evolution of more stable resistance in cancer. That was also in JTP. And if you're interested in this sort of drug-induced resistance and these treatment ideas I was talking about in the beginning, we wrote a book chapter on that recently, which is going to come out in the next months, I think. So stay tuned for that. Otherwise, thank you for listening, and I'm happy to take questions.
Thank you for your talk. So I had a question about the slide where you showed that the having the number of dead cells actually improves your estimate for certain of these parameters.
Yeah.
And I was wondering if you could speak a little bit more as to why that is. Is it just your system is underdetermined, and so adding in observations of another variable just helps us get more accuracy? Is there something, I don't know, a little bit more intrinsic to the parameter that you can estimate better?
Okay, so first of all, I should say that I didn't show a slide where, so in this example, I think we had 6 time points and 3 replicates. And like if you bring the number of replicates up to 100, then you can estimate the birth rates. Okay. So like all of the, theoretically, they're all identifiable, even if you don't add the extra data. Mm-hmm. But as for, if you can't do that, but you can estimate the number of dead cells, I would kind of remind you that in this experimental setup, you're starting out with isolated subpopulations. That's what we were doing in the numerical experiments. So like if you have an isolated subpopulations, you get measurements on the number of dead cells in the beginning. Mm-hmm. You're essentially seeing the death rate of the cell. And if you also know the net birth rate, which you already know, then you know the birth rate. Yeah. That's sort of the idea.
Thank you.
Okay.